From Microbe TV, this is Office Hours for Wednesday, August 14th, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet where you can come and meet with a virology professor and get your questions answered, and chat with each other, and be part of a nice community. Let's welcome our moderators tonight. I saw Les from California here. I saw Barb Mack from UK. I saw Steph from San Francisco. And I believe that's it for tonight. Thank you so much for moderating and keeping things civil. And welcome everyone. Tell us where you're from. We'll spend a little bit of time figuring out where you're from and shouting out for you. And then we have for tonight a few things, miscellaneous, but the big one is a mini lecture on MERS, Middle East Respiratory Syndrome. And this one is not present in my virology course. My course is mostly about process, not about individual viruses. So this is unique. So here, you can only find this here, this mini lecture here on Office Hours. I'm going to do more of these. I really like doing this. By the way, you have asked me like how many glasses I have. I had to organize them because they're getting out of control in my desk. I put them in a box, right? All different colors depending on my mood. Yeah, they're all cheap glasses from Amazon. You know, this is probably $20-30 for this basket here. But you know, they're made to look at a computer screen. So it's fun to do that. Now, one thing I wanted to point out here, this is very cool. I got a new mic stand. Basically, it's on the floor. It's sitting on the floor, as you can see, because the previous stand was attached to the desk. The top of the desk is a hollow core door, basically. And every time I would put my hand down on it, the sound would reverberate through the microphone. This is just ridiculous. And since I've been streaming here lately, I wanted to fix this. So I bought a mic stand for the floor, but it's a little bit tipsy. So I needed to weight it down, and you can see I used the microscope. This is, in fact, my father's microscope, who used to use it in his practice in the 60s. And I believe we have a listener from Coxsackie, New York, who I promised it to. It's yours. I think Alana is your name. I may be wrong about that. But I have a brick I can substitute for the microscope, but I thought it was fun to use that. So, okay. Where are you from? Let's see. What do we have here? Philip is from Wales, UK. Let me see now. I have a new system here. I'm going to have to move this up a little bit. Is that big enough for you folks? Is this font big enough? Do you want it like to be a little bit bigger? Edit font. Maybe it's 27. I think 30 maybe. Let's try 30. Just did 27. Okay. Okay. Philip is from Wales, UK. Rima is from Iowa. I have to move this because it's conflicting. So I have a new logo here, right? I decided we got a blue look because blue are the colors of Microbe TV, and I made a blue logo and some blue contributor information up here, moving up in the world. Okay, Rima is from Iowa. Lise is from Columbus, Ohio. Wayne is from Urbana, Illinois, where it's 83F28C. I'm going to make this a little bigger because it's hard for me to see. So it's, anyway, why did it default to 27? Let's make it, there you go, 36. Let's see. Yes, this says it's in Columbus. It's 81F in tomato overload. Yeah, you know that this defaults to the old size now. I wonder why. It's interesting. I'm going to have to figure out at a later date how to get it to default, because when I do it for each one, well, anyway, tomato overload, that happens, yeah. Abdul Aziz is from Saudi Arabia. Abdul, you requested MERS, you got it. I'm going to hear all about it. 32C. Karen's playlist is in northern Maine, where it's 66F with poor air quality because of the Canadian wildfires. You blame our Canadian neighbors. That's right. This is a very interesting name. Shen Shandy Beauty Fusion. Is that your business? I'm here from Tassie. Foggy moor, mild morning, post snow. Hello, everyone, from the little island down under. Tasmania, right? Oh, you got a beauty fusion? You got a beauty salon there? Cool. That's pretty neat. This is probably the farthest person from us, right? Will is in Changsha. 8 a.m., I guess. Boosters. We need boosters. What do you mean? Immune boosters or vaccine boosters? You can have both. We are releasing immune boosters pretty much every week. John is from Minneapolis, 71F, and cool. Kang is in Chicago, 25C and partly cloudy, but more clouds are to arrive after midnight. Nice. By the way, I'm a match freak now. I got red glasses. I got a red watch band. I have a red wire on my microphone here. And then my mouse pad is lined. The edge is a red stitch. I got red HDMI cables. And now if I change my glasses, I can't change all that stuff. They're going to have to be complementary to red. Chris from Montreal, big storm here heading your way now. Oh dear. So Friday, which was this previous Friday, 2:45, right? Coming up on Twiv, lights go out. Never came, well, they came back hours later. We had to cancel Twiv and TWIP, which we just did on Monday. I'll have some more words about Twiv in a moment. Kip and Laura, thank you for your contribution to science communication. The news with respect to Mpox, Clade 1B, my sister, nurse practitioner in Tennessee, is hoping the CDC monkeypox grant will be extended beyond December. Too bad money is an issue. So we covered this big outbreak in Africa on Twiv, which we recorded Monday, and it will drop Sunday. We had one scheduled for Sunday already, the long COVID with Judith Bruckfeld. So there was no skipping there. Thank you, Kip, for your support. Frank is in Santa Barbara, where it's 77. Spartacus is in the UK. I don't know how to say it, but I'm saying Spartacus. Visto, 16C in Sydney. Down under, Abdul Rahman is in Manhattan. Not too far. It's 38 miles from me, more or less. 50-minute train ride. But I'm out here in New Jersey tonight. Mark Martin! Yo, how are you, Mark? Tacoma, Washington. Nice to see you. I know Mark knows a lot of things, but you can even learn here. Do you know why I know this, Mark? Because I learn too. When people ask you questions, you learn, right? I love it. I love it. Questella, I know, is from Ohio. She's been here since like the beginning of this live stream. We started this live stream early COVID. It used to be called Q&A with A and V. Amy was my co-host, and then we transitioned it to office hours. Richard is in Portland, Oregon. Brian is in Montreal, 25C. Frank, actually Frank is another one of our moderators. You can tell by the blue little wrench next to his name. Frank is in New York, not too far from here. PGL is Patty, New Hampshire Seacoast. You like the blue layout? Yeah, so basically we have the video frame, and then behind it is a blue layer. It's actually got a gradient. You can see it kind of goes from light to blue. And I decided to use the micro TV color, which is blue, and put little white borders, just to make something a little different. Thanks. I'm glad you like it, Steph, because if you don't like it, then it's got to change. Doreen's in the Berkshires, which is like Western Massachusetts, right? Something like that. Claire is in the UK. Mike and Lori are from Seattle. Angela is in upstate Manhattan, New York City, like the north end, like 170s. Cheryl is from Santa Rosa, California. Yuxing, hello. I don't know where you are. Last time you were in China. Maybe you're still there. Airy Ozone from Asheville. I thought you'd like my little basket of glasses. So many colors. You know, it started out with just blue. Remember the apocryphal blue? And it just matches the Micro TV logo. I wore this for a long time. Then I diversified. But blue would go with the red stuff here, right? I could get a blue wristband, but the mic wire, I don't want to change every time. EG is from Mississauga, 25C. Rainy is in eastern North Carolina. What has become of Karen? Karen's at the incubator. Well, now she's probably home. But so you remember she used to participate when I was at the incubator. She was in person, and that worked nicely. So I haven't been to the incubator for a while to do these streams. So when I do it from the incubator, definitely have Karen back. Yeah, because I know you guys liked her. Midge is from Sydney, Australia. Pete is in London. Mild 18C, that's pretty cool. Rip de Comp is in Taos, New Mexico. Hi, Amy, if you're there. I don't know if Amy's here, but if you are here, Amy, say hello. Miss you. It's beautiful here, sunny after overnight rain. Use a microscope just like that. Yeah, it's a microscope that's fully functional. It's enormously heavy. It must be 20 to 30 pounds. Solid metal. It's from the 60s, and it was my dad's. He used to use it in his office. It's very cool. And I have offered it to Elena from Coxsackie, New York. I sound a bit over-modulated. Yeah, the volume is a bit high. Let's pull it back a little. I see it getting into the yellow. So, I'm going to bring it back 75%. Often people say they can't hear me. I've been tweaking my audio chain lately. And so I have more ability to control it than I had before. All right, is that better? Sounds a little fuzzy. Okay, I cut back the volume. Do you like that? Is Amy in the house? People are saying hello, Amy, but I don't see her. Okay, airy ozone. Artemis is in Montreal. Huge rainfall, remnants of Debbie last week. Yeah. Joseph is in Ontario. Mark Martin, giant viruses tomorrow on Matters Microbial. Excuse me. I also have a mute button, which I didn't have before. Looking forward to giant viruses. Yuxing is in Hangzhou. Okay, that's where you were the last few times. Laura is in San Francisco. I like the blue frame. Good. I'm glad you like it. So this streaming software I've been using for years. I never bothered to really learn to customize. I just turned it on and started talking. But it turns out you can do quite a few good things. And another one you're going to see today during my presentation. It's very cool. I've been working for you folks. I want to make your experience better because you learn easier if you have a good experience. Jerry is in Winnipeg, Canada. Martha is in Charleston, South Carolina. GSD Bio, that's Jonathan. And Sanitas, California, our favorite nephrologist, Carol from Bakersfield. Temperature finally under 100. Silent Runner, Wichita, Kansas, 95. Whoa. Frank wants to know if there are any COVID antivirals. Yeah, there are some being developed, but they're not quick to market because it's hard to test them in this environment, right? You would have to test them together with standard of care, Paxlovid, Molnupiravir, Remdesivir. So it's not easy to test them. And also, well, you could do a clinical trial and fix this, but people are doing home testing now, so you'd have to fix that. But obviously that can be done. Lucy is in Sechelt, BC, British Columbia. Jessica, Toledo, Ohio. I see what happens to the font now. It shrinks. The more text, it gets smaller and smaller. So actually, that's probably a, see, shrink. Text box. Is it dynamic? I don't want it to truncate. Let's try dynamic. It didn't make any difference. Anyway, Jessica from Ohio. Helen is from North Carolina. Can you talk about the cap for which the new HIV therapy drug is named lenacapavir? Capsid. Yes. Do I have a capsid? I don't have an HIV capsid here, but lenacapavir is an inhibitor of the capsid, which is the protein assembly inside the envelope that contains the RNA genome. And we talked about it on Clinical Update last week. Daniel. It's a novel target. It hasn't been targeted before. And they did a study, I forget where. I think it was in Africa. Twice pre-treatment a year, pre-exposure prophylaxis, twice a year. 0 HIV cases in the people who were treated. Pretty amazing. So that's lenacapavir, capsid. Bernard is in D.C. tonight. Did Vince talk about the new monkeypox? All right, so there's an outbreak of clade 1b monkeypox in Africa. We talked about this on the Twiv that will drop on Sunday. And it doesn't spread via cough. No, it's mainly via pretty close contact, just like the other monkeypoxes. So that's not correct that it spreads by cough. Oral Pouchet virus detected in Europe. Yeah, this is one that is emerging. It's coming to new places. Can you change the blue to red? Yeah, I could. But the blue is Microbe TV branding, right? It's all about branding. I sure we could change it to red, but we'll try it another time. Shen used to have a beauty business, but she hasn't been able to change her ID. Okay. She's retired. Well, if you were still working, I'd come to Tasmania and you could cut my hair. But you're not. Wasim is in Kashmir. Whoa. Wow. Started listening to Twiv in 2008 as a postgrad in veterinary virology. Well, first of all, I don't think we've had someone from Kashmir before. So welcome. But I guess this is your first time here on the stream. This is value added to the Microbe TV universe. You're going to learn new things. It's cool. Thanks for coming, Wasim. Theodore is from Greece. Cascadia News Service is a great name, but I don't know where you are from. Oh, here we go. From Point Roberts, Washington, 71C. Thank you. That's an interesting name. Razor is in Saskatchewan, 30 degrees C. Hat is in the San Fernando Valley, where it's 90 F. Wendy is from Pleasantville, New York. Does everyone ask you if you're a Padawan? Right? I'm sure they do. I saw someone whose last name was Sephora. And I said, is that your store? And she said, I wish. Looking at people's name tags is a great way to break the ice. And most of the time, they really like it. So last year, I went to give blood at a clinic in Manhattan, and the lady did a nice job. And I looked at her badge when I left, and I said, thank you, and her name. And she touched me on my shoulder. She said, thank you. These simple things, just saying people's names, it's great. It's not going to work all the time, right? But it will. It can. People are saying hello to Amy, but I don't see her. Trevor is in Nashville. Thanks for your help with my microbiology degree in college. That's great. Thank you, Abdul, for your super chat. By the way, if you want to support science communication at Microbe TV, just below the video window, there's a button with a $sign. Yeah, US centric. I know you can push it and you can put in an amount. It converts to your currency. Or up here, you can go to microbe.tv/contribute or Venmo. You can support us. Microbe TV is a 501c3 nonprofit. Your contributions are federal, U.S. tax deductible. Okay, better on the sound. Thank you. Patricia, we read your letter on Daniels, of course. Folks, if your letter's not getting read, just ask me. You can do it here. You can send me an email. I'm happy to help. The key is brevity. You don't have to be one sentence, but some folks send in a big, 8.5 by 11 full of text. It's kind of long. And I would have to end up truncating it, and I don't always feel comfortable doing that. So if you can make it shorter, max half a page. So that's what Patricia did. She got her letter in. So thank you very much. Appreciate it. Shen is Tasmania, 9 degrees C. Wow. Hello, fellow Australian listeners. Originally from Lake Macquarie, north of Sydney. Yeah, we have at least one. Visto, maybe others. The Vincent R. blue box could be located at the top left. Yeah, I know, but lower thirds are... I could move the text. How about if I move the text more to the center? I get a little thing saying when it's in the center. The lower thirds are supposed to be at the lower third, right? But tell me if you want me to put it at the top, because it's a matter of me grabbing it and just moving it up here. It'll be upper thirds. It's non-standard, but tell me if you like that or not. I'm listening, folks. I'm listening. Mini lectures. Rima wants to know how my hiccups stopped. They just stopped. Actually, towards the end, during the mini lectures, they basically stopped. They resumed a little, and then towards the end, they went away. And then that was it. No more, and I have no hiccups since then. So thank you very much for asking. Spartavus. It's a shame we don't get to see Amy that often anymore. She has a lot of character and used to make me smile. Now, there's no question that Amy has a lot of character, for sure. And sometimes she comes in the chat, but she really can't participate. Martha, if no one has made a home for your father's scope, I'd be happy to pay the shipping and provide a revered location for it in my home. Well, I'll keep that in mind. Alana from Coxsackie, New York, had first dibs. I will find out if she still wants it. Maybe, it's a year ago. Maybe she finally got her own scope, in which case I'm happy to send it to you. Hello, Vanity Nutrition. Welcome. Actually, it looks better with the chat by itself on the bottom and the information stuff at the top, right? It's a good idea. You have to be flexible in this world. Rima wants that microscope. I tell you, this is it's my dad's. My dad was a physician. He went to medical school in Italy, so he was born in Italy. Went to medical school in Italy, and then came to the U.S. in 1950 and trained for 10 years to be a thoracic surgeon. He did an internship, a residency, a surgical residency, and a thoracic surgical residency. It took 10 years. 1960, he opened his office, and he had that microscope in it. He only practiced for 24 years. Sadly, he died in 1984. But he was an excellent surgeon, I hear. But it was a different era, right? Ancient. No PCR, no molecular medicine, nothing. They made things work. All right, let me just take this question. The nervous system connected to the lungs. Yeah, of course it is. I mean, cough reflexes and they have to go through the brain, right? You have a local irritation that goes to the brain, then the brain says the muscles cough. Totally. Everything's connected to the nervous system. Yeah, as Patricia says, anything with muscles is connected to the nervous system. But sensory, so your skin, right? No muscles in the outer layers, the living cells of your skin, but they're innervated sensory so that when you put your hand near a flame, you pull back so you don't hurt yourself. Shen, once a stylist, always a stylist. Your most welcome 45-year career had 25 staff. I travel with my scissors everywhere. I was blessed to work in Papua New Guinea for 4 years. Well, if you ever come to New York, bring your scissors. You can come to the incubator. We'll set up a chair, and you can cut my hair just to see what you can do. I mean, it's not so bad. I kind of like it, right? But it's always interesting to try something else. Yes, Padawan. Padawan were the Jedi in training, and your Padawan is close. Nobody ever said that to you? Interesting. Oh, they ask if their family was from Padua. That's a good one, too. Privyet, Elena. Reno, Nevada. Privyet. Welcome. My pleasure, Jonathan. I have knowledge I want to share. What's the point of keeping it to yourself? I learned a long time ago that it's rewarding to share knowledge with people. And of course, technology gives us this great vehicle, right? That you can share it with a lot of people. I'm not just standing in a lecture hall at Columbia University and sharing with just, a few people, but I'm sharing with all of you all around the world, right? I mean, look where people are from. Kashmir, Tasmania, oh my gosh, China. My pleasure. All right, the name at the top. Okay, everyone likes it at the top. Very good. Amy has brilliance and we love her humor. Absolutely. Yes, so Amy did a clinical update with Daniel when I was not around, and that was fun. She got a one-off. Yep. Shen says your hiccup stopped when you started laughing as you were about to try an alternative treatment you were not sure of. Then you giggled quite a few times and your hiccup stopped. So keep laughing. Oh, the different treatments that people offered to me last time. That was amazing. Thank you so much. Yes. Have you seen people who turn old microscopes into table lamps? No, I haven't seen that, but yeah, it might. Sure, it makes sense. You could put the lamp in the. You take out the eyepiece and put the stem of the lamp in there. That'd be cool. Neat. So in Princeton, I stay at the Palmer House when we go to write our textbooks, and in the lobby there's a microscope lamp. Yes, I'll take a picture of it, Mark, and show you next time. It's very cool. It's very cool. Let's see, who else is here? Lenacapavir was only tested in a small group of women. They're actually transgender women. We did look at the study. I looked at it. It is small, for sure, but there were no HIV/AIDS cases in the treated group, and there were in the untreated group. But I agree it's small, and it needs to be further tested. This is not licensed, right? So it needs to be further tested, but it's promising. Yep. Okay. Now we got through where everyone is from. Okay, one thing I wanted to talk about just briefly. Now let's see if I can use some cool stuff here, some cool technology. So hang on a sec while I go to this website. I should have had this. All queued up. Hold on. Boy. Okay, here we go. I'm just going to share my screen. So this Dan Wilson's website, of YouTube channel, of course, and the title of his video is "The Problem with Vinay Prasad," and so many people have written into Twiv to say, "Can you please have Vinay Prasad on Twiv?" And we say no because he's out there, he spreads misinformation. But Dan likes to do these debunkings, which we don't really do on Twiv, with one exception, COVID origins. So I would send you to this. This is really good, and you can see why I don't want to have him on, okay? I wanted to share that with you. One more thing before we get into a mini lecture. We're going to do a delay on the Twiv releases from now on. So we're going to record on Fridays as usual, and typically we release on a Sunday, but we're going to release the following Sunday. But there will be episodes released every Sunday. It'll just be from the previous week. And we've already got that in sync. I released the long COVID episode on Sunday, and so now we're in sync. Now, the reason we're doing this is because production on a weekend is hard, right? So I have a video editor who spends all of Saturday doing Twiv. I used to do that, and it's a pain. And even though he's willing to do it, I'm not sure that he's going to do it forever. And who knows, things may change. And so I want to be prepared. So he's now can be able to edit it during the week and post it on the Sunday. So you're not going to notice really very much because it's going to be papers as usual. Maybe the news will be a week delayed, but I don't think you're going to notice that. So that's what's happening with Twiv. Okay? All right, now time for a little bit of mini lecture. So watch this. I can do this. I click a button. I almost click a button. And I have a mini lecture ready to go. I was always fumbling with this, right? Setting it up in real time, which is stupid. I don't have to fumble. I've got my little face in the upper corner of the screen. I've got my slides here, and I can move my cursor and talk to you. Okay, MERS, Middle East Respiratory Syndrome. I want to first talk a little bit about emerging viruses because that's what this is, right? What's an emerging virus? Now, this is a little bit from the course, but it doesn't hurt to repeat things, right? An emerging virus is the causative agent of a new or previously unrecognized infection. So 2 possible definitions. This is a term coined in the 90s, but of course emerging viruses aren't new. We just have been able to discover them. Since the rise of agriculture, 11,000 years ago or so, new infectious agents have invaded human populations because they can be sustained by numbers that were unknown before agriculture and commerce. We lived in hunter-gatherer packs, few hundred people, and that doesn't sustain infectious agents. But as soon as we learned how to do agriculture and more and more people could live together, getting food, then population numbers grew and infectious agents begun. And as I said, we've recently gotten really good at detecting emerging viruses. A little more definition: an emerging virus can have an expanded host range with an increase in disease not previously obvious. For example, poliomyelitis for much of human history was a rare disease, never caused epidemics, and then around 1900, boom, epidemics. So it's a increase in disease, and understanding why is of course important. Now, sometimes, or often, every time there's a new virus, it comes from an animal. And a virus goes from a wild or domesticated animal to a human. It's called a zoonosis. The zoo, the animal part, it's a zoonosis. And sometimes these spillovers from animals to humans establish a new virus in the population, a new human virus. For example, SIV moving from chimps to humans, that's now HIV-1, a human virus. Or SARS-CoV-2 from bats to humans, that's now a new human virus. But not all crossovers, not all emerging viruses establish themselves as human viruses. Often, cross-species infection cannot be sustained. For example, every time there's an Ebola virus outbreak, it's a new spillover from bats or some other animal reservoir to humans. MERS coronavirus, as we'll see today, typically new spillover from camels to humans with limited human-to-human transmission. Now remember, Ebola virus in West African outbreak 2015, over 20,000 cases, obviously a lot of transmission, but it was a special case because the transmission mainly occurred in hospitals and at homes. So there was no community transmission, people walking around spreading it in theaters and sporting events and restaurants like SARS-CoV-2. It doesn't happen with Ebola virus, doesn't happen with MERS-CoV-2. Now, I love this graph. Ancestral origins of human pathogens. So these are 4 bars on this bar graph. Now, the y-axis is the number of viruses belonging to genera. So these numbers in here are genera of viruses, not species, but a little higher order, genus. And then on the bottom are the different categories. So we have 31 viruses, members of 31 genera that are human viruses, but they came from animals. So poliovirus and herpes simplex virus, all these human viruses, HIV-1, they came from animals at some point. Members of 37 genera are zoonotic pathogens. They continue to spill over from animal reservoirs into people. Ebola virus, Nipah virus, even West Nile virus. It doesn't become a human virus. It doesn't transmit from human to human. It goes from mosquito to human, and the mosquito gets it from a bird or a rodent reservoir. Now, the next one, 16 members of 16 viral genera. We got from ancestors of Homo sapiens, right? The hominid ancestors. They got the viruses from animals, and then they passed it on to the Homo species. And then finally, members of 6 genera, we acquired from our Homo relatives, Homo sapiens from Neanderthalensis and Denisovensis, is that the right way to say it? Denisovans. They got it from animals and they passed it on to us. Every virus came from nature. And if you want to ignore history, well, then it's your demise because the facts are all there. Okay. Now, why is this that viruses emerge? And let me move this little thingy here. Overriding factors driving the emergence of infectious diseases. Well, number one is population. Look at the population growth. Billions on the y-axis and years on the x. Look at this incredible growth. Population growth everywhere, invading all ecosystems. And this makes us contact animals which have their viruses, right? That's just the way it is. What else? Well, anthropogenic activities. Humans continuously devise new ways to encounter viruses. Dams, water impoundments, irrigation, deforestation, transport of animals, blood transfusion, air travel, urbanization, daycare centers, hot tubs, air conditioning, used tires that carry mosquitoes, climate change, natural disasters. Look at that. Look at this snapshot of all the flights in the air at any one time. Amazing. So we have devised ways to encounter new viruses. Don't blame nature. Blame humans because we set it up. Okay. Finally, it's important to understand that evolution plays a big role in the emergence of pathogens. Because as viruses multiply in their hosts in nature, whether it's a bat or a pig or a duck or whatever, they mutate. And so virus populations are quasi-species. They're composed of millions and millions of mutants in any one animal, many, many different mutants, one of which may randomly be able to infect a human. And should that virus encounter a human, then it could start a pandemic. But many viruses exist that could infect humans, but they never encounter humans. Now, SARS-CoV-2 existed somewhere in a bat, and it was brought into an animal market and contacted humans. So we facilitated its contact with humans by bringing in animals from the surrounding area to Wuhan that had been in contact with bats, which had the virus. And then in the meat market, it spread to humans. So you have lots of mutants existing in the animal reservoirs, and those mutants allow adaptation to new hosts and environments. That's how evolution plays a role in emergence. Okay, that brings us to MERS and MERS-CoV. MERS. So there's two names for this. Just like HIV/AIDS, just like COVID, SARS-CoV-2, MERS, Middle East Respiratory Syndrome, and MERS coronavirus is the virus. The disease is MERS. MERS-CoV is the virus. You know, when COVID and SARS-CoV-2 were first named, people were like, oh, why do we have to have two? Well, just look at what's been happening in history, for God's sakes. We do that all the time. Don't complain. So the first case ever seen of MERS, 2012 in KSA, a 60-year-old male patient died of pneumonia and renal failure. They suspected virus infection, recovered virus from this patient. They sequenced the genome immediately. They identified it as a new coronavirus, a novel coronavirus. They identified the receptor for this virus, dipeptidyl peptidase 4, which interestingly is not present in the human upper respiratory tract. Okay? So that's going to tell you right away why this virus does not transmit well among people. If a virus is to transmit among people efficiently, it has to reproduce in the upper tract, because that's where you're shedding virus all the time. You don't shed a lot of virus from the lower tract. You know, when they tell you to give me a really deep cough, we want mucus from way down in your lung. It's hard to do, right? So without upper tract reproduction, it's hard for a virus to spread extensively. Now, it doesn't say that it won't change at some point. It could change receptors, perhaps, but it hasn't. So this virus was rapidly identified, sequenced, and found to be closely related to bat coronaviruses, although there is a phylogenetic gap. This we'll see in a moment what I mean. And it's not SARS-CoV. It's distant from SARS-CoV. It's a different virus because, of course, as soon as they saw it was a coronavirus, people started to worry, but it's not SARS-CoV. So in terms of the origin, two kinds of bats carry coronaviruses similar to MERS coronavirus. The polymerase, RNA-dependent RNA polymerase of MERS-CoV has 92% amino acid identity to the pipistrelle bat HKU5, and the S and the N genes are more closely related to a different bat virus, HKU4. But it doesn't prove that these are the ancestors of SARS-CoV-2. It just says they share a common ancestor. Now, more recently, an interesting virus was discovered in South African Neuromycia capensis bats that has 85% similarity with MERS-CoV. Very, very close. And it's called NeoCoV. Doesn't cause MERS, but it's close. And we'll talk about this in a moment. Well, right now, this paper published in 2022, which I think we did on Twiv, this virus, NeoCoV, close relative of MERS coronavirus, right? NeoCoV, South Africa, it binds ACE2, not DPP4. So what is that telling you? This ACE2 we know is in the upper tract. SARS-CoV-2 binds ACE2. It spreads really well because it reproduces in the upper tract. So it's possible that NeoCoV could eventually emerge as a pandemic MERS virus, but that hasn't happened yet. But other things have to be in place as well. But the first step, having the receptor in the upper tract, is pretty important. Okay, so here's a phylogenetic tree of some coronaviruses to put this in perspective. So again, this means how they're related on their genomes. So down here is MERS coronavirus, and it's close to some bat coronaviruses that I just told you about, HKU4 and 5. And then a good distance away is SARS-CoV, and a good distance away is SARS-CoV-2. About 60% identity with SARS-CoV-2. So they are quite distant. And down here you have the human common cold coronavirus. So MERS sits on its own. It's quite a different viruses from SARS and SARS-CoV-2. Now, what I haven't told you is that humans get the virus from camels, and a specific kind of camel, the dromedary camel. I'm going to talk to you about that in a moment. But at the moment, we see three clades of MERS coronavirus. There's clade A, B, and C. And clade B mainly is in the Arabian Peninsula, a few other places. It's the most diversified, and it's found in humans and in camels. Clade A, Saudi Arabia, Jordan, and UAE. Again, humans and camels. And clade C in other countries, Egypt, Morocco, Ethiopia, Burkina Faso, Nigeria, and Kenya. But most of the MERS originates in the Arabian Peninsula. There's no MERS in Nigeria or Kenya. The virus is in camels there, but there's no human MERS for reasons that we don't understand. The only human MERS is in the Arabian Peninsula. Everything else is exported from there, travel related. So the virus is spread from dromedary camels to humans. So we think that some years ago, maybe hundreds of years ago, the virus went from a bat to a camel, established itself in camels. I'm going to show you some evidence that camels have been infected for a long time. And then it infected people starting in 2012. Unfortunately, not all infections have a camel source. Many do, but not all of them. There's some spread from person to person in hospitals and within households. There's no community spread. There's none of this where the virus is shed for days before symptoms and you can get it in mass gatherings and so forth. So we don't know why those cases have no camel source, but many of them do. Now, just to put this in the perspective of the Coronaviridae, right? The Coronaviridae family is divided into 4 genera: alpha, beta, gamma, and delta coronaviruses. And the viruses that we're talking about, MERS and SARS-CoV-1 and SARS-CoV-2, are all beta coronaviruses. And then you see some of the other species of coronaviruses are gamma and delta coronavirus, the human common cold coronas. Some of them are alpha coronas. Okay, so that's where these lie: MERS, SARS-CoV-1 and 2, beta coronaviruses. Oops, I went to a link. So as I said before, MERS, the disease, is only reported in the Arabian Peninsula and in travelers from this region. There's no MERS in Africa or Asia, even though they have infected dromedaries. Okay? There's no MERS disease. There have been 2 cases of MERS in the U.S. They've both been imported. However, in one study in Nigeria, camel abattoir workers were shown to be seropositive for MERS-CoV. So they weren't sick, but they had antibodies to the virus. So this is telling us that if you have close contact with camels, which you do in an abattoir for camels, right, you can be infected. But why it doesn't cause disease in Africa, we have no idea. I mean, it could be genetic basis, slightly different clade of virus. We just don't know. And it would be nice to find out, because understanding what controls infection and disease is important, right? So some of the signs and symptoms, this is a very typical severe acute respiratory disease. Most people have a severe respiratory disease, symptom onset 5 or 6 days after exposure, fever, cough, shortness of breath. Risk factors. There are a variety of diseases: diabetes, cardiovascular disease, chronic lung disease, heart disease, kidney disease, cancer, or recent travel in or near the Arabian Peninsula, or if you work in a healthcare setting in that area, especially if you had direct physical contact with camels, touching or grooming, or had close contact with a person who is sick with fever or respiratory symptoms. So these are all different risk factors for MERS. So basically, the CDC is not telling you not to go to the Arabian Peninsula. This is a very rare disease, but you should be aware of the risk factors. So here's an example of the incidence of MERS. So these are confirmed cases of MERS by place of infection and month of onset. This is from 2012, when we first discovered it, to July 2024. Been 2,622 cases, including 953 deaths. It's about a 35% case fatality rate. And you can see by the colors, most of these are in the Middle East. So here is 2012, when we first discover the virus, and there's quite a large outbreak in the Middle East, which eventually subsides mainly because of information and control measures. You know, we say camels are a risk factor. If you're going to work with them, you have to take precautions. But outside of the Middle East, you see mainly in 2015, big outbreak actually in South Korea. An infected traveler from the Middle East went to South Korea. He got sick. He was admitted to a hospital. They didn't know what he had. They didn't do any infection control. And so he infected a lot of other people in the hospital until they realized what it was. This is a epidemic curve of the cases in KSA from 2012 to 2023. And you can see cases are in blue, peak in 2014, and slowly reducing as we got more aware of how to mitigate this exposure. And the deaths are quite high as well. But again, this is where the vast majority of cases occur. In fact, all the index cases occur. Everything else is a travel-associated case. Nobody gets MERS in any of the other countries where camels have the virus. This is an older map, but I like it because it shows you the transmission in geographic range. Okay, so camels transmitted to each other throughout northern Africa, the Middle East, parts of India, Afghanistan, Pakistan. But the transmission from camels to humans and limited human-to-human transmission, largely in the Arabian Peninsula. And these red lines are exported cases to different countries. People who were visiting the Arabian Peninsula got infected and then went on elsewhere. There's no local acquisition of this virus, say in the U.S. or in different parts of Europe. So this is the dromedary camel. There are different kinds of camels and camelids, as you may know. I think let's move the. I can't move it. And here there's the Bactrian camel, domestic and wild, the dromedary. Then we have the guanaico, llamas, vicuñas, and alpacas, camelids. And these colored areas are the ranges of these animals. And the one we're concerned with is the dromedary camel. This is the only one that is endemically infected with MERS coronavirus. And you can see its range, northern Africa, Arabian Peninsula, and moving towards India. There's a little bit of, there are a few dromedary camels in Australia. They were brought there, actually. And there's actually a feral population of wild dromedary camels. But they're not infected with MERS coronavirus in Australia. It's a cool animal, right? It's just really neat. I like the way they look. Anyway, a number of studies have looked for seropositivity in camels. Either antibodies, some cases they looked for RNA by PCR, and samples were available back from 1981 through 2020. So you can see samples of camel serum. These are dromedary camel serum. From 1980s are positive, are antibody positive against MERS coronavirus. So this virus has been circulating for many years, probably even longer than this, probably over 100 years in camels. And either it never infected humans or we didn't notice it. And then suddenly in 2012 it starts. But you see, antibody positive, these parts of Africa, interestingly, Kazakhstan, and then RNA positive, showing an ongoing infection most likely in Arabian Peninsula and parts of Africa. But again, the disease only occurs in the Arabian Peninsula, not in Africa, not in Kazakhstan. Very interesting. Now, here's a nice study, because you may be asking yourself, why does this happen all of a sudden? You know, the camels have been infected for hundreds of years with MERS coronavirus. Why in 2012 do we first see this? So a study was done in Qatar to try and understand the drivers of MERS emergence. Here's a reference for this here. It's done by Marion Koopmans and her colleagues. So here's the story. Basically, you know, Qatar became a wealthy country. Oil and gas exploitation led to an increase in the gross domestic product, growth in population over the 30 years, lifestyle transformed from Bedouin to urban sedentary life. You get increased obesity, you get other comorbidities, which are risk factors for MERS. And so then, in this wealthy environment, camel husbandry flourishes. People race camels, they have them as pets. And this exacerbates the desertification, that forced banning of free grazing. Camels are housed in compact barns alongside their workers, facilitating interaction with the people. And so this transition in husbandry leading to high-density camel farming, together with increased exposure to humans, increased camel movement for the racing and breeding industry, It's a convergence of factors driving spillover of MERS-CoV from camels to humans. It's our fault, folks. We did it. We moved camels around, decided they were cool to do things with, had more contact with camels. We got their virus. It's our fault. Here are two graphs that show this. Here's in Cotter, the increase in GDP over time from the 80s to the present. You can see it's going up. It's in US dollars. And the camel population is going up. And these are the total number of camels in this gray line. And then there's meat camels and milk camels. So obviously the vast majority are used for other purposes like pets and racing and so forth. So that's an interesting take on why suddenly you have this disease where the virus has been around for years. So it doesn't tell us why it's not present in Africa, though. All right, so what can we do? So, there have been less than 3,000 cases total. Very hard to do anything for that. There are lots of experimental vaccines and antivirals. Shoutouts, the chimpadenovirus vector has been used to carry the spike. mRNA vaccines encoding the spike of MERS-CoV, vaccinia virus vaccines, and many others. I mean, every kind of vaccine has been tested. None of them have been approved. It's not clear who would get it. There aren't that many cases. Maybe immunizing camels would be one way, a one world health approach, right? So the camels get infected at birth in their herd, and they don't get very sick. They get a little respiratory drizzle, very little lower tract disease, and it passes through the herd. But immunizing the camels has been proposed, but this is not a sterilizing vaccine. It's not going to prevent them from shedding. It's, in my opinion, not going to prevent transmission to humans. But how do you test that, right? So that's a tough one. So basically, no vaccine is approved. And there's some antivirals that we know, remdesivir, molnupiravir, they work against the virus in animal models. They haven't been approved. They haven't gone through clinical trials. I mean, the other problem is, how do you do a clinical trial? There are not that many cases, right? There are monoclonal antibodies that could be used therapeutically, but it's hard to test them because there aren't that many cases. So we're in a point where the only thing you can do is supportive therapy for MERS. So this summarizes now all the coronaviruses, where they came from. This is very cool. Here we have SARS-CoV-2, right? Which originated in a bat, probably some animals at the Huanan meat market, seafood market, civet cats and raccoon dogs, into humans. And in MERS, bats to camels to humans. SARS-1, bats to civet cats to humans. And then all the 4 common cold coronaviruses, some kind of animal, bats or rodents, through an intermediary to human. There's even a coronavirus of pigs, severe acute diarrhea syndrome coronavirus, which went from bats to pigs. Do you see the pattern here, folks? There's no need to say this came from a lab. In fact, there's no evidence. All right, so that's MERS. And if you want more virology, I have a whole virology course on YouTube. There's the kind of unwieldy link, but you'll find it there. Thanks for listening, folks. Back to me. Let's see if we have some questions. We did have questions before, so I'm going to start answering those. Ah, yes, there's some good questions here. Let's start at the top. Scroll our way down. Monkeypox, Autopuche, Kashmir, Cascadian News Service. Oh, here we go. From Rushby, thoughts about polio in Gaza. Are there other viral outbreaks that may be of greater concern for them? Yes, of course. So the problem in Gaza, right, is that there's no sanitation. And there's polio circulating. It's in wastewater. There haven't been any cases of polio yet, but it's inevitable because vaccination is lagging, right? But other viruses, well, so any virus that's spread in sewage, right? Noroviruses and rotaviruses, which can be severe if you don't have the ability to hydrate, can be a real problem. But then the respiratory viruses as well, because people are living in crowded, unsanitary conditions. Measles would be the first one, right? Because it's so infectious, and vaccination rates are lagging, right? Because it's a mess. Other respiratory viruses, influenza viruses, SARS-CoV-2, respiratory syncytial virus, basically any human-human transmission is going to be a problem. And hopefully they can address this and improve the conditions and start vaccinating people and maybe prevent this from happening. And I know they're going in and trying to vaccinate against polio. They should also be doing that against measles and other viruses as well. Carol says, my 91-year-old mom got COVID vaccine last September, then another one in April. Will she need coverage with the new vaccine? So your mom is old and susceptible to severe disease. Now, the physician will probably say, get it. I'm not going to make a recommendation, right? Because I can't give you medical advice. But immunologically, only within maybe the first month or two after immunization will the vaccine prevent even mild disease. But she's going to get infected, and that's not good in a 91-year-old. So the plan should be to, if she can take Paxlovid, to take it, or if not, one of the other antivirals, as early as possible to improve her chances of survival. And that's Daniel Griffin's. He would say, get vaccinated and be prepared to use Paxlovid. I don't think it hurts to get vaccinated. Angry Penguin. I read that antibody-dependent enhancement has been observed in dengue infection. What is antibody-dependent enhancement? Well, I could actually bring up a slide because now I know how to share my slides really well. So let me do that. So let's see. That would be in pathogenesis lecture. So that's going to be in the second half of the course. Is it going to be infection basics? No, it's not going to be that. Innate and intrinsic? Nope. Is it? This is adaptive. Well, let's see. Adaptive immunity, no. So it's going to be 15, which is mechanisms of pathogenesis. There we go. All right. So I'm going to show you the dengue enhancement slide. Here we go. It's perfect. Play. And I'm going to go to my screen share button. Look at that. It's like I don't have to do anything. So in dengue, there are 4 serotypes of virus. Let's say you get infected with serotype 1 and you recover and you're going to make antibodies, right, against serotype 1. And now let's say later you get infected with any of the other 3 serotypes, 2, 3, or 4. You're going to make a memory response to the first serotype you saw, in this case, serotype 1. So antibodies from primary infection. These will bind the serotype that infects you, but it will not block infectivity because it's the wrong serotype. However, the antibody bound to the dengue virus will bind to receptors on cells like monocytes here. It will let the virus get in that cell where it normally doesn't. It'll make more virus, and you will trigger inflammation. And so that's a more severe disease than dengue itself. It's accompanied by hemorrhage and shock and a high fatality rate. So that's antibody-dependent enhancement, right? Because the antibody is binding the virus and it's enhancing its ability to get into a cell that it would not normally infect. A monocyte doesn't have a receptor for dengue virus, but when the virus is bound to an antibody, there is a receptor for the antibody on the surface of the monocyte. So there you go. Good question. And lets me demonstrate my technology here. Is it normal to get mild scar or slight pain on lower lung after COVID? Yeah, I don't know. So what do you mean? Like scarring on x-rays? Or so even in mild COVID, you can get ground glass opacities on a chest x-ray or CAT scan. Okay, I guess it's a CAT scan that shows that. But you guys can correct me. Right, so that can happen. In fact, the very first family in Wuhan that was infected, the parents got sick, and they actually did a CAT scan of the kid. He was not symptomatic, but he had ground glass opacity. So that can happen. I don't know about slight pain, if that's common. The lungs don't have nerves, so you're not going to feel pain from this scarring unless bad things happen. So I'm not slight pain. I'm not sure about that, but the ground glass, yeah, for sure. See, John writes, there are two clades. We have ACAM 2000 and Genios with fewer undesired side effects. TPOX, we have two antivirals and public health communication. Yes. So what we're trying to do, what WHO is trying to do, is get the vaccines and antivirals to the outbreak areas. They have to be approved, right? They have to be given emergency use authorization because they are not so far in those countries. And so someone has to pay for it. The U.S. and other countries are paying for it. They have to be approved there and then distributed. So we do have means to control the outbreak. I mean, this is really unfortunate. The outbreak has been going now for several years. And yet, until it got out of control, nobody wanted to do anything. That's what WHO is for. I don't know why. So someone asked the question about mitochondria. Mitochondria are fascinating. Integration and gene flow in both directions between nucleus and mitochondria. A co-opted bacterium. Yeah, mitochondria are bacteria that came into some ancestor of our eukaryotic cells and stayed. They stayed. How does it go? Come for the... I don't remember. Pete, who suggested I move the name up, says, I was in print and amateur publishing from 77 to 88. Can still see graphic design. We put out an underground environment mag in the UK until 84. Thank you for the suggestion. I kind of like it. Yeah. And don't be surprised if I change everything to red to match my glasses sometime and my watch band and my microphone cable. But I do like microphone cables, especially colored ones. So don't be surprised if I buy a blue one. Oh my gosh, I'm out of control, right? Any plans to visit San Diego? Next year, maybe. ASM Microbe is in Los Angeles. And I think that's June. And, I don't get to the West Coast very often, so I will probably do an extended stay. In fact, I think I would like to hit San Francisco, LA, and San Diego. So Stay tuned. Stay tuned, yeah. Mm-hmm. Oh, Renzo, hello from Brampton, Ontario. I haven't seen you for a while. True or false? Truth or dare? As long as COVID test is positive, the person is infectious. It's false. Not necessarily. You could be positive and not be infectious. So you could be antigen positive. So let's say antigen because that's what we're doing these days, right? And then you feel better and it's negative, but then it could turn positive again because there's some antigen just sticking around. Remember, when you do a nasal swab, it's not an accurate science, right? You're getting different amounts of stuff, different parts of your nose each time. You know, maybe one time you miss the infected cells and another time you get it. So, no, it does not mean you're infectious. And people can be positive for many, many days, even weeks, but that does not mean you're infectious, no. Debunk the funk. Yes, so Dan Wilson is Debunk the Funk. Does a very good job counting things. He's cool, calm, and collected. He tells you why someone is spewing misinformation. Yes, and Vinay annoys Patricia a lot. Yeah, he's annoying because he makes bad conclusions and he overinterprets data and he shouts just to get people to listen to him. You know, if I spewed misinformation, I could have 1 million followers. Why would I do that? I'm a teacher. I want to teach you the truth. No. I mean, it doesn't make any sense. Maybe he thinks he's doing the right thing, right? But he's not. M asks, can viruses, bacteria, germs be suspended in typical soap solutions? Wonder if soap can be contaminated but yet still be effective as we understand the mechanism of hand washing. All right, so this is a little involved question here. So it depends on the virus, right? So poliovirus will retain infectivity in salt. All right, for sure. So you may say, why do I wash my hands when I go to the toilet? Well, it's actually, if you do it long enough, the physical act of scrubbing your hands and rinsing will get rid of most viruses. To reduce the quantity so it's no longer a problem. And other non-envelope viruses like polio can survive in soap. But envelope viruses like SARS-CoV-2, influenza virus, respiratory syncytial virus, their infectivity will be destroyed by soap. And so they will not contaminate you. Now, norovirus is another one that will resist soap as well, and ethanol. So that you have to wash your hands for not just 2 seconds, right? Recite the alphabet because you need to physically remove everything or most of it. Yeah. I don't have a new mic. I have a new mic stand, but I reduced the gain because I have a new interface that has more control over what I can do. Yeah. Okay. I just clicked something I should have. Non-virologists should stay. Yeah, I mean, everyone should stay in their lane. He's not staying in his lane. I've always said if you want to understand viruses, ask a virologist, right? So he's not a virologist. He's a medical doctor, and he has a different specialty. So he figures out that he just thinks he can figure everything out. Well, I admire that you think you can figure things out. And often scientists can by reading and talking to people, but he doesn't do that. He draws his own wacko conclusions. Yeah, here's the episode on Prasad that you might want to listen to. Why do some cross-species infections sustain themselves and others don't? Well, I think the difference, the answer is multiple. There are multiple answers. Let's take MERS, which we just talked about, right? So humans don't have the receptor for the virus in the upper tract. That's a game killer right there, right? Because if you can't have upper tract infection, you will never transmit it unless you're really close. So I think that's possible, that transmission is inefficient. For Ebola virus, that it's not a case of receptor, but we don't know. Again, maybe it never adapts to humans to be able to transmit successfully, which requires a certain level of replication. And, if you look at viruses, some adapt and some don't, right? HIV adapted, poliovirus adapted, influenza viruses, SARS-CoV-2. So we study them to try and figure out why. SARS-CoV-2, long incubation period with shedding, without symptoms, so you can get community spread, receptors in the upper tract, very efficient reproduction. So those are the key factors, but not all. I mean, you have to also antagonize host defenses, but not so that you overwhelm the host. I think there are a lot of issues. Rio says, I would like to recommend Dr. Malhotra, a cardiologist, as a guest. He did a 2-hour interview 2 weeks ago on Diary of a CEO. His experience during COVID is unique and changed over time. Well, I would have to look into Dr. Malhotra as a cardiologist. I'm not a cardiologist, I'm a virologist. And he or she doesn't have something to tell us about viruses, right? I mean, Diary of a CEO is a perfect place, but I would need to be convinced that this is an interesting topic for a virology podcast. Now, we could make other podcasts that don't require that, right? I've been toying with a career in health and medicine type of podcast, right? Talk to different people in different fields about their careers, and I think that would be a perfect place. Now, you say that the unique COVID experience is unique. I'd have to look at that and see exactly what. I don't want to Prasad, right? For sure. Visto says, so many human viruses. Rats must say humans are so dirty. Rats have their own viruses too. Hot tubs. Yes, you notice that on the slide of risk factors for spillover, right? So hot tubs are sources of viruses because people don't properly sanitize them, like adenoviruses. If you ever get a pink eye after you've been in a hot tub, they don't have enough chlorine in the hot tub. So they're a source of spillover. Well, hot tubs didn't exist X number of years ago, and humans decided, we need to have hot tubs so we can soak and relax in the hot water. And now we introduce another source of viruses from other people because their viruses go in the water and it's not sufficiently chlorinated to inactivate infectivity. That guy was a guest on Camel, this Dr. Malhotra. Okay, well, that's bad. I like the picture of the camel. Thank you, Rita. And yes, KSA is Saudi Arabia, Kingdom of Saudi Arabia. Sorry about that. I thought everyone knew. So Kang wants to know, is the infectious dose more related to the binding kinetics of virions, like when more H5N1 is needed to infect humans? I mean, infectious dose in people you're talking about. How much virus you need to infect the host, a person, let's say humans, right? That's what you're talking about? Huh. I think it's multifactorial. I think it has to do with how susceptible the cells are to infection, whether, if there's no receptor, forget it, right? Like we talked about with MERS in the upper tract. H5N1, there's no receptor in the upper tract either. It has to go down into the lower tract. So you need to inhale a good amount of virus down into that lower tract. But I think it's also how much virus is produced so that it can sustain the infection, how well the virus antagonizes immune responses. So I don't think it's just binding kinetics, no. I think that's one of the factors. If you have weak binding, you may need more virus, but not necessarily. I wouldn't assume that, but it's a possibility, yeah. Yes, you should not kiss your camel on the mouth. But actually, you don't have to kiss your camel to get infected. You just have to be near it because they're breathing and the aerosol is going to get you with MERS coronavirus. Oh, good. We'll like this lecture. Every journalist reporting on health matters needs to listen to and get educated. So, I mean, the key, you see what the key questions are, right? They're very straightforward. And it's like, why doesn't it spread? Where did it come from? Why is it not a problem in countries where camels are infected? And what led to the emergence? I mean, all these are interesting questions, right? But yeah, this is a half hour. What's the problem, journalists? I'm too busy. I don't have time. I bet you spend a lot of time on TikTok, though, and YouTube. So there's this business where I don't have time. I just never buy it. You can always make time for something. It's a matter of desire. Thank you. I'm glad you like it. Can you publish the mini lecture as a separate video? Yeah, sure. These streams are recorded. I could get the video and simply edit out the rest of the stream. Yeah, I think that would be cool. I will do that. It's a good idea. Right, folks? That's a great idea. Should I write it down? Do I have anything to write on? Yeah, I have stickies. Stickies. Do I have a pen? Of course. Does a bear. Okay. MERS mini lecture. Actually, I should do that with some of the others too, like the polio one and some others, because this is unique. It doesn't exist, right? It's cool. John says Vinay got promoted by Breaking Points, a channel that promoted Lab Leak, demonized Dashak, and itself promoted by Joe Rogan. Debunk the Funk is a good takedown of Vinay this week. Yeah. That's why I wanted you to that's why I recommended it. And I used to watch out for all these weird, sketchy places promoting other people. They don't promote us. We never got promoted by Breaking Points. Why? We tell the truth. They're not interested in the truth. There are camel beauty contests. There was a scandal when some camels had cosmetic surgery. Oh my gosh. I wonder what you would do to improve a camel's looks. A tummy tuck, facelift. Oh my gosh. Crazy stuff. Oh, apparently Dr. Malhotra is an anti-vaxxer. All right, that's it. Dromedary is a cool word. I like dromedary. Yeah, it's got all the right syllables and the right accent. Yeah, you're absolutely right. Rima visited Saudi several times and my heart got stuck there. You like it, huh, Rima? Never been. One day. Yeah, I'd love to. Love to go and do some pods with local scientists. I think that would be a blast. So we're arguing about why Dr. Malhotra should be given a chance. And Rio says he's been pro-vaccine, but changed his stance on the COVID vaccine. Well, that's enough to say no. Why would you change your stance on the COVID vaccine? What do you think you know that no one else does? That the mRNA vaccines cause myocarditis? We know that. That the vectors cause blood clots? We know that. Don't tell me it integrates into your DNA. It's BS. So this is just wrong. So COVID vaccines saved many, many lives. So that disqualifies him. And, just seeing things differently is not good enough. You have to have it supported by the science. People hide behind this. They get defended about this differently. Oh, I'm different and people don't like it. No, you're not different. You're just spewing misinformation. Okay. Wonder if Paxlovid works on MERS. Yeah, it does. In the paper where they described Paxlovid, it does inhibit in cell culture, but we don't know in animals, right? If it reaches the right concentration and so forth. So that would have to be done. And as far as I know, it hasn't been done. I heard Visto says, I heard camelids have some special mini antibodies. So camels have single chain antibodies. Yeah, I'll show you. I mean, my newfound ability to show you stuff. Here, single chain antibody. Let's find a good picture. Everything is now damned AI and Google. All right, let's see if we can get a picture. Yeah, they're really cool. That is this a good one? And maybe that's a good one. Okay, yeah, this is good. Okay, beautiful. Let's go to the screen share. So it's a little small, but so this is a normal antibody. It has two chains, right? Two heavy and two light chains. And camelid antibodies typically have single chains. So here is a single chain antibody, right, where just one heavy and one light chain. So it doesn't have both of these antigen combining sites. It has one. And the reason that's interesting is because you can make these in bacteria. So that's great because you have less issues with folding of the protein. You can select for high affinity nanobodies, and and you can combine different antigen binding parts. You could basically make designer antibodies at will from the sequence. You don't have to go through lymph nodes and affinity maturation and all that. So they're very cool. Now, does that have anything to do with MERS and spillover? No, I don't think so. I mean, the camels are just infected with MERS coronavirus, and they make antibodies to it, they control it, and so forth. Yeah. Apparently, Mark Twain has a camel story. There you go. Thank you. Any idea where bats first contracted coronaviruses? Hundreds of thousands of years ago, probably when bats first emerged, right? They've been in bats for ages. They've been in bats for ages. Just like viruses have been in other animals as well. So, yeah. Razor says, may I ask a simple question? How does the camel's immune system derate the symptoms, and can that research perhaps be applied to the human immune system? All right, so you're asking why camels have mild disease when they get infected with MERS-CoV, right? It's the same as why many people who get infected with SARS-CoV-2 develop mild disease. They may even be asymptomatic. Actually, there are people asymptomatically infected with MERS coronavirus as well. So, because we know in populations we can see antibodies, and people have never been sick. It's a good question. I think it's a combination of factors. I think sorry, I got distracted by a question. I think it's how robust the immune response is. I think the over exuberant immune response can cause severe symptoms. I think early control of infection by, say, interferon can help make fewer symptoms. Essentially, we don't know for any virus what modulates the symptomatology. And a camel's not a really good animal to study that in because they're large and expensive, and you would rather do that in smaller animals like mice. Artemis wants to know, should getting shingles count as a vaccine dose? Well, if you actually get it, right? If you've got overt shingles with a rash on your skin and you know it's shingles and nothing else, I don't see why it's an issue. Sure, I don't see why but I had shingles in 2008 and I got 2 shingles vaccine doses because it doesn't hurt. And you just don't know in a person, everybody's different and you just don't know how you responded to infection. You really don't know. And so I would be saying you may not have made a good immune response to the infection. So I would just err on the side of caution and get 2 shingles vaccine doses. Thank you, John, for your contribution to science communication. Why do you think there's so much COVID-2 around? Just people being completely normal and very little mitigation? This is an interesting question. We talk about this all the time with Daniel on Clinical Update. Why is there so much SARS-CoV-2? It's a peak in cases everywhere. And it's July, August, right? It's obviously a very different virus because there's no peak of influenza right now. Influenza is going to peak in the late fall and winter. And so will probably SARS-CoV-2 again. So there's something physically different about the SARS-CoV-2 virus that enables it to transmit very effectively in the hot and humid summer months. I think part of it is perhaps that people are hanging out with each other in the summer. It's been a hot summer. Daniel suggests it's a hot summer. People are inside in the AC, and those are places where you could transmit viruses. But it just doesn't seem right. There's got to be something more than that. And it must be the physical properties of the virus make it capable of transmitting in multiple seasons, whereas I don't think influenza transmits very well, at least in temperate climates, in hot and humid environments, although it does in the tropics. So that's another story altogether. So there must be something different about the SARS-CoV-2 virus particle. What happened to the viewer number that used to live in the lower right-hand part of the screen? I don't even remember seeing that. I'm looking at the stream now. I don't see it. Is it a what do we call it? Is it a preference? No. I don't know. Good question. I don't know where it is. Anybody know about this viewer number? Well, anyway, I can tell you right now there are 150 people here and 125 likes. So please hit the like button. Just help attract people here. Yeah, Hepe and Leishmania. That's right, for Gaza. Thank you, Rima. War seems to be a common theme with regards to viral disease transmission. And you can't see the number or the like button. I don't know where that would be. Let me see if I go to the YouTube page, right? Because I'm on my own little special interface here. Yes, I see that it's live. It shows you the number of likes. I don't know. Good question. I don't know. Maybe someone else can figure it out because I don't know. I like when people get excited. Oh, yeah, antibody dependence in dengue. Great question. Hope that you like the answer. Spamlet wants to know, is there a test I can ask my GP for as a vaccinated person to tell if I have been infected with SARS-CoV-2 virus itself? Yes, you can ask for a antibody test that will detect antibodies to the N protein. Okay, so the vaccines all have spike, so you're going to have antibodies to spike. But you will have antibodies to N only if you've been infected. So ask your GP to find a test that will detect anti-N antibodies. So Gary has COVID for 5 days. Light coughing. I feel 100% still test positive. Am I infectious still? Maybe or maybe not, but maybe at 7 days you won't be. So I would just, if you want to go somewhere, wear a mask. It's hard to know because we don't assay infectivity, and the correlation is really poor between the positive tests and infectivity. Will, as a 78-year-old, I got a second infection of COVID in March, 1 month after my booster. Do not rely on vaccines to prevent infection. Get jabbed, wear your N95. I agree, they don't prevent infections. That's what I was saying before with the 91-year-old. Just get ready with a Paxlovid because they're going to get an infection. In a 91-year-old, any infection is not good. Paxlovid. These vaccines, we're changing them to match variants, but I'm not sure that makes any difference. You're still going to get infected, and you're always going to be protected against disease because the T cells hit cross-reactive parts of the virus that don't change in the variants. So if you want to get boosted, that's great, but you should still have a plan to take Paxlovid because you're going to get infected. Vanity. Put exposure. I think I didn't recognize you earlier as a moderator. Thank you for moderating. Could exposure to high levels of non-receptive and non-permissive virions elicit any type of reaction? I can see how ingested ones wouldn't, as they'd go right through, but what about inhaled? So, yes, you can get some immune response because obviously that's how vaccines work, right? Inactivated vaccines don't replicate. You make an immune response. But you'd have to have a high amount of virus, and I think that's very atypical. But in theory, you could get a response, yeah. Wild idea. Would getting measles clear out dengue antibodies? Yeah, getting measles. So if you got dengue and then you got measles, measles would wipe out your dengue memory, so you would not get ADE, antibody-dependent enhancement. So this is a theoretical question because, I don't know how that would work. Very funny. Ground glass opacity does not sound good at all. Actually, many people have ground glass opacities, right? And you do ground glass opacities are abnormal areas of hazy opacification that appear on chest x-rays or CT scans of the lung. They can be caused by fluid displacing air, airway collapse, fibrosis, neoplastic process, infections, congestive heart failure, alveolar hemorrhage, interstitial lung disease. So you often see them when you're having a CAT scan for some other purpose. Oh, you have ground glass opacities in your lungs. So it can be indicative of a pathogenic process, but they often go away also. Early on in Wuhan, they changed from testing to CT scans, which gave clear indication. Of course, CT capacity is not huge. You can only do so many CT scans a day at any hospital because you don't have that many machines. It takes half an hour per patient. So it's not a mass screening thing, but they used it well. Yes. Solution to dengue antibody dependent enhancement: get infected by all four types. Well, get vaccinated with a vaccine that has all four serotypes, right? And there is such a vaccine. So if you're in a place where you're at high risk for dengue, just do that. Problem solved. Vaccines solve problems. Besides bacteriophages, are there other viruses being considered as therapies? John, to look at the last lecture in my virology course. Therapeutic viruses, not just phages, but all kinds of eukaryotic viruses, adeno-associated virus, adenovirus, measles virus, VSV, pox viruses for not just vaccines, but killing cancers and curing monogenic diseases. So yeah, viruses are going to save us all. What does ACE do on cells of the lungs? So normally it's involved with blood pressure, right? I don't know. It's a good question. So ACE2 is expressed on 60% of human cells. And I wonder if it has a function in other cells. Let's take a look. Let's look this up. I'm curious. So ACE2, the homolog of ACE, acts as its physiological counterbalance, providing homeostatic regulation of circulating angiotensin II levels. ACE2 is a zinc metalloenzyme on the surface of cells. So I think it can regulate homeostasis on any cell type, right? So that's what it would be doing in the lung, because obviously it's not there as a receptor for SARS-CoV-2, right? No love for the Pacific Northwest? Who said that? I like the Pacific Northwest. Where'd you get that from, Mark? First of all, I don't ever diss any place, but I like the Pacific Northwest. Maybe I'll visit next year. Abdur Rahman says a patient had a positive HBV core antibody test, negative antigen, and negative DNA. Well, if you have antibodies and no protein or DNA, this infection's been cleared, right? That's what I would say. Anthony just finished up Richard Preston's book of Hot Zone and discovered your channel. Well, you didn't discover through Hot Zone because there's no way that Preston ever mentions us. But thank you for finding us and welcome, Anthony. And Will has a list of expert idiots, and Vinay is number 46. How many are on your list, Will? Why do most viruses originate from tropical or temperate climates? Well, what's left? Really cold climates. Not much living there, right? And not many people, not many animals, so there's not a lot of opportunity for spillover. You know, temperate, us, tropical, yeah, I mean, that's most of the world except for the frozen zones. Current world numbers for TB, I don't know. But, you know, we could ask Dr. Google. TB numbers. See if it figures that out. In 2022, 10.6 million people had TB. 1.3 million people died. 6% were in people with HIV. 46% Southeast Asia. 23% Africa. 18% Western Pacific. In the same year, the U.S. had 8,331 TB cases. That's a lot of TB, right? If you do not have water, will just rubbing your hands together vigorously clear viruses? No. No, you have to have the cleansing fluid, soap to dislodge, and the water to wash it away. Roman baths were original hot tubs. That's right. You know, they were big community hot tubs. Probably transmitted infections even then because there was no chlorine, right? Why is there no generally available bathroom device to reliably clean under the fingernails? Most people don't even have a nail brush, but most of them don't get under nails anyway. Well, first of all, you keep your nails short, right? I mean, I cut them often because the recipe for dirt under your nails is long nails. And I have a nail brush. It works really well. You know, you got to soap it up because the soap plays a big role and you need to scrub it hard. But if I'm working in dirt and I get some dirt under my nails, I don't want it under there. Boom, go into house, nail brush. So I think nail brushes work. Are some hot tubs chlorinated sufficiently? Yeah, for sure. Not most are, but now and then you get ones and you get red eye. Yeah. Huh. And one time, Mulholtra said vaccine killed 1 in 1,000 users. Luther says, do you know a good video to explain transcription and translation? Oh, I do, but I don't have the link to it now. But it's on YouTube. There are lots, actually. If you want, I can look it up. We may have it linked for in our textbook, actually. So let me take a look at that. All right, everybody likes the mini lecture series as a standalone. Okay. Bristo says, my mini lecture should be compulsory for journalists. They're not going to listen to me. They want to listen to Neil deGrasse Tyson. Well, I have nothing against Neil. He's a great astrophysicist, great communicator. But he doesn't, well, I think he probably does talk about viruses at some point, right? And so the journalists think, well, here's a star. I should listen to him. Oh, Aerosol, sure, conjunctivitis from hot tubs, but more problematic is the spread of Legionnaire's disease. That's correct. Water sources. Colombia's president just resigned. Wow. I didn't know that. Well, I'm not surprised because of the dust up over the campus protests in the spring. Is Osterholm's information good? I think he has reasonably good sources, yes. And he's mostly okay, but he does make some errors. He's way off on the variants, SARS-CoV-2, transmission, virulence. He just doesn't get those. But he does have some good information. Thanks for the straight talk. Thank you. It's the only talk I know how to do. Abdur says camels are getting fillers, Botox, hormones. They'd tie rubber bands around jowls and ears to trim them up. They crack down on it. Oh, that's terrible. Oh. Okay, 56. We need to wrap up. Let's see what we have here. The Plague by Albert Camus predicted a lot of the behaviors we saw resulting from the fear during COVID. Interesting. I'll have to read it. I didn't, and this is 1947. Yeah. Wow. Thank you. Spamlet says dromedary means running or racing, hence hippodrome, velodrome, etc. So what did they call Arabian camels before they started racing them? I don't know. Are you going to tell us? You got 194 people watching now, 150 likes. Hit the like, folks. WHO announced global health emergency. Why is it different? In fact, it should have been last year because it was spreading last year as well. It's been spreading for many years. The outbreak has been going on for a while. And it just got into the rest of the world, a couple of years ago that we all heard about it, but it's been ongoing and it should have been dealt with earlier. I agree. That's what I was saying. Likes are on your screen. Viewer number on the lower left. Okay. Okie dokie. Is taking the flu shot mid-August useless? Yeah, I think you should do October, November. I don't know if it is. I think it's too early in August. Yeah, totally. See, this is the thing with Ostrom. He said he got the XBB.1.5. He's going to get KP2 or KP3. Why? What's the evidence that they make any difference? There isn't any. Get Paxlovid if you get infected and you're of a certain risk. Don't keep getting boosters every 4 months. As Paul Offit says, it's ridiculous public health strategy. Where do we vaccinate people every 4 months? It's just that we don't understand what's going on, so we just default to having people get vaccinated every 4 months. Take Paxlovid if you get infected. Ground glass opacities also occur in mycoplasma pneumonia. Thank you very much. Abdur. Where's Abdur's question? I'm not purposely overlooking questions. I don't know which question it is, but is that Abdul about the Hep B? I did. So Will has over 60 people on his idiot list. Okay. There was an episode of House of a senator running for re-election with Hep C, and House wanted to give him Hep A to clear. No! House is not always right, You can't clear Hep C with Hep A. First of all, Hep A has its own issues. You never give Hep A or a vaccine even. It's not going to do anything. No. Nowadays you give antivirals for Hep C. They work very well. If the permafrost melts, will new viruses emerge? Maybe, because there are some viruses frozen. So it's possible, but we don't know what their host range would be. We know nothing about them. What should we do? Should we take lots of cores and figure it out? You could. There have been limited numbers of cores doing core drilling and sampling, yeah. Something in-depth on either Hep B or C. So you want a mini lecture on Hep B or C? So I have a couple of requests from last time. We could do Hep B and Hep C and then start to make videos of them independent. Yeah. You guys are telling me about the Pacific Northwest. I love it. I've been there, and I have no problem with it. I don't know why Mark said I was no love for it. Yes, Osterholm is an epidemiologist. He's not a virologist. Thank you, Steph. Absolutely. Stay in your lane. Do you think other countries perform biological warfare research? No, I don't think they do. The Soviet Union had a big planned program for many years. This flop, because you can't engineer new things. You can only take old things, and it's very hard to weaponize viruses. We have the best weapons we need, right? Bombs and so forth. You don't need viruses. It's a dumb thing to think about. And so if anyone's working on it, you know what the problem is with biological warfare? You're going to kill yourself. Oh, yeah, we'll make an antidote. Not likely. Not likely. Yeah, so DeGrasse could bring other experts on. But what? He's a smart man. He knows what questions to ask. I can imagine it would be okay. And then, of course, he's got a big reach, so it's good. So, but the journalists can't have him on to talk about science, virology. No, they need the experts. Okay, I answered it. There's the virology course list. Okay, Patricia, I answered it. All right, I get thorny if people say you're ignoring it, but I try not to ignore anything except things that are obviously offensive. Elizabeth says most developed countries have signed on to some weapons bans. Yeah, terrorists, not nations. So, probably maybe Russia's doing biological warfare. Who knows? But it's not going to work. It's not going to work. Don't do it. I wish the microbe TV community consulted on House. Junior, a new revision. It could be great. Dixon did some consulting for House. Yes. Hello from the Philippines, Talia 7. Yes. Osterholm is also a public health guy. You know what, folks? That's the end of the list. Thank you all for coming. I want to thank our moderators tonight. Let's see if I can remember. We had Les, we had Tom, we had Steph, we had Frank, we had Vanity Nutrition, we had Barb Mac UK. Whoa, 6 moderators. Thank you so much, moderators, for keeping it civil. Really appreciate it. Had 190 people at one point. Thank you so much for coming and listening. We'll make mini lectures a regular part of this. We'll release them separately. Do you want to know when the next office hours will be? It'll be Wednesday, the 28th of August. No, that can't be right. That's 2 weeks from now. Today's the 14th. Yeah, the 21st. Let me mark it on my calendar. By the way, the 21st of August, we have a Twivo live stream at 1 p.m. And then we have this office hours at 8 p.m. I'm trying to get Dixon on. We'll try and get him next week. I couldn't confirm him for this week, but many of you wanted him, so I'll try and get him next week. But as you see, we do a great job even without guests. Oh, one more point here. Anthony wants to know about USAMRIID. So we did a TWIV at USAMRIID, and TWIM is going back to USAMRIID to learn about their bacterial research. So you might want to listen to that. They were very honest with us. I didn't think they were hiding anything. All right, folks, thank you all for coming. Really appreciate your participation. And until you come back next week, please be safe. Thanks, everybody. Good night.